and the most growth.
It wasn't a fish.
So I decided to focus my experiment or my study on that microbe. So I took that microbe, and through successive streaking, I was able to isolate colonies of the microbe itself. And I then performed some phenotypic categorization or characterization tests here. In figure 3, you can see a motility test. So to get this, I use my microbe, which I call KSCIL,
Yeah, so when you start up
to make a liquid culture, choice septic broth. I then placed 50 micrograms of that culture on the center of this plate, which is 0.3% choice septic agar. And this is about 24 hours after that, so you can see that it spreads out from the center location, which suggests that the microbe is motile. I also performed a gram stain test with my microbe, and I found out that it is gram positive. This image is at 1,000 times magnification. And you can see that it might be kind of hard to see, but you can see that each microbe is around maybe 3 to 10 micrometers in length. They are gram positive, they're rod shaped. I also performed an antibiotic resistance test. As you can see here, it's a little hard to see, but the results of this test are in this table. So I basically put a liquid culture of my microbe all over this plate, and then I put 5 different antibiotics on there on these little discs. And I measured the distance of inhibition of growth to see what it was resistant to and what it wasn't resistant to. So I found that it was resistant to penicillin and ampicillin, mostly ampicillin. So that's very interesting because people are drinking this and it is antibiotic resistant, so it could potentially be pathogenic and hard to treat. So after I categorized it phenotypically, I also wanted to look at the genetics of it. So I isolated the DNA, extracted it, and then I sent that to the UNH Genetics Center, Hubbard Genome Center. And they sequenced everything fully, put it on their server RON. And I looked at that server and got the entire genome. And I trimmed it using a program called Truematic to make it a little bit neater, a little bit easier to see. And I then configured all the sequences of DNA into contigs and annotate it using Proca. So that was able to let me look through the genome and find certain genes. So knowing that, I was able to extract the 16S rRNA gene, and I put that into a BLAST N search to try to figure out what it was. And that resulted in a match with Bacillus subtilis. I had a 99% ID score, but since it's a Bacillus, the 16S rRNA isn't very good at actually identifying it. So I looked at a couple other housekeeping genes to see what I might potentially have. So I looked at the DNA-directed RNA polymerase subunit beta gene and the adenyl succinate synthetase, which I researched online, found that they're both usually used to classify bacilli. So both of those matched with this organism called Bacillus koshi. The matches were all right, 86% for the DNA-directed RNA polymerase subunit beta and 84% for the adenyl succinate synthetase. They're not great, but usually 95% is like the cutoff. But they came up with Bacillus koshi, and there are a few other genes I looked at that also came up with Bacillus koshi, so I was thinking at that point I might have Bacillus koshi. So after I figured out that might be Bacillus koshi, I looked at some specific genes based on the morphology and just the phenotypic traits of my microbe. So since I found it in river water, I found it in the river water in late January, so the river was very cold, so I figured the microbe probably had cold shock proteins. So I looked for cold shock proteins in the DNA, and I found this cold shock protein CSPD gene, and that had a 96% ID score after I did a BLAST N search, and that came up with Bacillus koshi. Then I also looked for a swarm motility protein gene, and I found that also matched with Bacillus koshi, but it was only a 69% match, and I also had a 69% match for the beta-lactam inducible penicillin binding protein, and that is associated with penicillin resistance, so that's why I looked for that. So I had an alright idea that I might have Bacillus koshi, but I wasn't sure because these aren't very good match ratings. So there were some other microbes that also consistently came up, and I didn't include those in this table just because it doesn't really go with the idea of what I might have had. But these other microbes, Bacillus oceanissediminis and Bacillus wernickeyi, they came up fairly consistently too. So I compared the genome sizes on this and found that my microbe up top, which had genome size of almost 5.8 million base pairs, was significantly different than both Bacillus kochi and Bacillus reinekii, and it was kind of similar to Bacillus oceanis. But if you look at the GC content, it's also fairly different than most of these, where, again, it's the closest to Bacillus oceanis, but it's still 1.8% different in the guanine cytosine content. And then after performing an average nucleotide identity search, or comparison, I found that Bacillus kochi had the most in common based on nucleotide data with my microbe, but it was only 72% roughly, so that's still very far off. And to actually identify it as Bacillus kochi, I would ideally have a match rate of around 95%. So down here in figures 8 and figure 9, you can see the data in this table graphically represented. So this is comparing the cumulative length of the genome of my microbe with Bacillus kochi, which is the reference screen in this figure. So the red line is KSE-IL my microbe, and then this dotted dashed line is Bacillus kochi. So you can see that my microbe has significantly larger genome than Bacillus kochi. And then down here, this figure 9 compares the guanine cytosine content. And again, the red line is my microbe, KSE-IL, and the dashed line is Bacillus kochi. So again, you can see that my microbe has significantly more guanine cytosine content, as it's this little bit to the right. Then I also compared the number of protein coding sequences as well as the number of rRNA genes and tRNA genes of my microbe with these same 3 microbes. I found that, again, it was fairly different. Oceanis again, was the closest in terms of protein coding sequences. But if you look at the rRNA, it's pretty different. Oceanis has almost or more than twice as much rRNA coding genes than my bacterium, even though tRNA genes are still fairly close. There's only a discrepancy of 5. So what I conclude after this is that I can't actually identify the microbe I found as Bacillus kochi. It has some characteristics in common with Bacillus kochi, but after comparing the average nucleotide identity, there's no way that I can definitively say it is Bacillus kochi. Due to its antibiotic resistance to penicillin and ampicillin, it's hard to say whether or not it's pathogenic, but it is important to know if it is pathogenic because it might potentially be hard to treat. It's also resistant to iodine. As I mentioned, that was my protocol to isolate it. So hikers could be drinking this water potentially. That's another reason that you might want to know whether or not it's pathogenic. So future directions would include phylogenetically classifying the microbe more fully to try to figure out what it might be, to find out more about genes it have that might be involved with pathogenicity, as well as to find out if maybe it could potentially be a novel strain or even a novel species, since it has so many discrepancies between anything else I was able to find.